Good day, everyone. Welcome to our lesson in biology. Today, we will be talking about cellular respiration. But first, what is cellular respiration? As we can read here in our screen, cellular respiration is a set of metabolic reactions and processes that take place in the cells of organisms to convert chemical energy from oxygen molecules or nutrients into adenosine triphosphate and then release waste products. As we can see here in our screen, the chemical equation and chemical reaction is flashed, and that's basically the basic principle of cellular respiration. There are two types of pathways in which ATP can be generated from pyruvate molecules, a product of glycolysis. Those are aerobic respiration and anaerobic respiration. Cellular respiration can occur both aerobically using oxygen or anaerobically without oxygen. During aerobic cellular respiration, glucose reacts with oxygen, forming ATP that can be used by the cell. The location or the main sites of this respiration in aerobic, it is in cytoplasm and mitochondria, while for anaerobic, it is located in cytoplasm. And that's it for our lesson, and for my part, and it will be further discussed by my groupmate.
Hello, good day everyone. So now I'm going to discuss the aerobic respiration. So when we say aerobic, it means with air and so needs oxygen. Aerobic respiration releases energy in cells by breaking down food substances whilst in the presence of oxygen. It is represented by this simplified equation. Glucose is broken down by oxygen to release the byproducts of carbon dioxide and water, and energy is released, which is then used to make a special molecule called ATP. ATP is how energy is stored for later use by the body. And aerobic respiration happens all the time in all cells, usually in mitochondria. That is why, because of the process, the mitochondria is referred to as the powerhouse of the cell. Take note that in aerobic respiration, there is a complete breakdown of glucose molecule. As the glucose molecule breakdown in aerobic respiration is complete, therefore it releases a lot of energy. Again, you just need to remember that glucose is broken down in the presence of oxygen to release carbon dioxide, water, and energy as byproducts of the reaction. So, aerobic respiration involves two phases. These are the Krebs cycle and electron transport chain, which will be discussed by the next reporters.
Good day, everyone. So this time we were going to be discussing about the Krebs cycle and how does it work. The tricarboxylic acid or TCA cycle, also known as the Krebs cycle, is a main source of energy for cells and an important part of the aerobic respiration. A Krebs cycle is a central pathway that provides a unifying point for many metabolites which feed into its various points. It follows 8 steps. The first step is when the acetyl coenzyme A 2-carbon molecules joins with oxaloacetate, a 4-carbon molecule, to form a citrate or the 6-carbon molecule. Next step is when the citrate is converted to isocitrate, an isomer of citrate. And the third step is when the isocitrate is oxidized to alpha-ketoglutarate, a 5-carbon molecule, which results in the release of carbon dioxide or CO2. And also in the formation of one molecule of NADH. The fourth step is when the alpha-ketoglutarate is oxidized to form a 4-carbon molecule. This binds into coenzyme A, forming succinyl coenzyme A. A second molecule of NADH is produced as well alongside the second molecule of carbon dioxide. The next step is when the succinyl coenzyme A is then converted to succinate, a 4-carbon molecule and 1 GTP molecule is produced. Sixth step is the succinate is converted into fumarate, a 4-carbon molecule and a molecule of FADH2 is produced. The seventh step is the fumarate is converted to malate, another 4-carbon molecule, and lastly is when the malate is then converted into oxaloacetate, and the third molecule of NADH is also produced. The primary rule of Krebs cycle or TCA cycle is the production of NADH and FADH2, but it also produces molecules that supply various biosynthetic processes.
Greetings everyone, I am Joseph G. Mardeno here to discuss to you the electron chain transport. So in this process, as we can see, it says a group of compounds that pass electron from one to another via redox reactions coupled with a transfer of proton across a membrane to create a proton gradient that drives ATP synthesis. So in this, we can see in the figure, there are 4 protein complexes, of which in the first complex we can see that is where the electrons will be fed through, of which the first components, these proteins facilitate a series of redox reactions shuttling the electrons downhill to one component to another, with each structure having a higher affinity of electrons than the last. Yet, it is not the creation of ATP, but rather a byproduct, or rather a generation of a proton gradient across the membrane. So, the protons accumulate outside, or rather in the intermembrane space. In this case, it will power, or rather be fed through the ATP synthase, of which, as is self-explanatory, will create the ATP that we need. So, in short, as the ATP, or rather the electrons from the intermembrane will pass through the ATP, create the ATP synthesis, which creates the ATP, or this process is also called the chemiosmosis. That is all. Thank you.
Anaerobic respiration. When oxygen is not present, glycolysis is followed by a different pathway. The combined process of this pathway and glycolysis is called fermentation. Fermentation releases energy from food molecules by producing ATP in the absence of oxygen. So, anaerobic respiration is the type of respiration through which cells can break down sugar to generate energy in the absence of oxygen. This is contrast to the highly efficient process of aerobic respiration, which relies on oxygen to produce energy.
Thank you, reporters. And now I will be discussing to you about the 2 types of anaerobic respiration. First of all, we have alcoholic fermentation. So, alcoholic fermentation is a process accomplished by yeast, some kinds of bacteria, or a few other microorganisms to convert sugars into ethyl alcohol and carbon dioxide. Alcoholic fermentation as well begins with the breakdown of sugars by yeast to form pyruvate molecules, which is known as glycolysis. So, as we can see here in our slides, the example here is bread, but we can also use wine as an example. So next, let us proceed to its steps or the formula. Alcohol fermentation has 2 steps. First of all, it has glycolysis and NADH regeneration. During glycolysis, 1 glucose molecule is converted to 2 pyruvate molecules, producing 2 net ATPs and 2 NADH. And lastly, we have lactic fermentation. Lactic fermentation is produced from carbohydrate materials as lactose in whey by the action of any of various organisms, but especially the lactic acid bacteria. It is also a bacterial process that takes place during the production of numerous food products. It provides the final products with characteristics like aromas and textures. It also plays a crucial role in food safety and hygiene. So the process of this is that lactic acid fermentation is a metabolic process by which glucose or other 6-carbon sugars, including disaccharides of 6-carbon sugars, examples of this is sucrose or lactose. They are converted into cellular energy and the metabolite lactate, which is lactic acid solution. So that would be all for our report, and thank you guys, and once again, good morning.